sharing her recent data and amazing work. And please, if you can share your screen and start.
Will do. Okay, can you see my screen?
Yes.
Excellent. Okay. Well, thank you so much, Christina, for the introduction and for the invitation to come speak with you all here. It's really wonderful to be able to be a part of this transatlantic speaker series. So thank you very much. So I'm going to start with the sex difference that really motivates my lab's work. And that's this one here that shows, so this is a figure from an Alzheimer's Association annual report showing the estimated lifetime risks of developing Alzheimer's disease at ages 65, 75, and 85, with men in the purple and women in the gold. And you can see that at each age, women are at greater risk of developing Alzheimer's disease than men. And we think this is due, at least in part, to, that's interesting, to menopause, to losses of estrogens in particular during menopause. And just to remind you, in adult women, estrogen levels are quite high, even though they fluctuate during the course of the menstrual cycle, but they are quite high during the reproductive years. And then early in the 40s, estrogen levels start to plummet. And so in women in their 50s and beyond, estrogen levels are quite low. And this is significant for the brain, and particularly for Alzheimer's disease, because neurons in the hippocampus and the prefrontal cortex in particular, so these are cognitive brain regions that are affected early in Alzheimer's disease, these brain regions, these neurons become dependent on estrogens as neurotrophic factors that help keep them functioning properly. And we think that the loss of estrogens at menopause actually puts these neurons at increased risk of neurodegeneration due to age-related changes, as well as neuropathological changes that occur as part of Alzheimer's, the Alzheimer's disease pathology. And so this would seem to be a fairly simple problem to solve, that if declining estrogen levels lead to increased risk of Alzheimer's disease, we can reduce that risk by replacing estrogens. But as I'm sure you're aware, estrogen therapy has its pros and cons. So, and that's because there are estrogen receptors in tissues throughout the body. So while there are definitely beneficial effects of estrogen therapy, particularly on bone strength and cholesterol profile, and definitely in terms of reducing hot flashes and other menopausal symptoms, there are documented, at least here in the US, clinical trials have shown small but statistically significant risks of various kinds of cancers, breast and uterine cancer, as well as heart disease and stroke. And so what we have been trying to do is to understand how the potent estrogen, 17-beta estradiol, which you can see the structure here, how does estradiol act on brain regions like the hippocampus to form memories? Because we think if we can understand the molecular and cellular bases underlying estrogen's effects on memory, that perhaps we can design treatments that provide the benefits of estrogens on cognition without some of those side effects. And so if we ask the question, how do we reduce memory dysfunction in older women? I'm going to cover three different bases today. So our first step is to try to understand how estradiol regulates memory in young animals. So how does it do this in an optimally functioning system? And we've done this work in both young females as well as males, but I'm just going to focus on females today. I don't have time to talk about the male data. And I'll spend probably most of my time in this space. And then we'll move on to the next step, which is understanding why hormone loss leads to memory dysfunction in models of menopause. So this would be aging animals as well as Alzheimer's disease. And I'll talk today about some of our newer data in an Alzheimer's mouse model. And then finally, once we have that information, how do we develop novel avenues for treatment? And I'll end by talking about one novel treatment in particular. So we have historically focused on the hippocampus as a locus of estrogen's effects on memory. This is just a number of views of the hippocampus and the rodent brain. It's a bilateral structure. In humans, it sits down here in the medial temporal lobe, but in rodents, it extends much more dorsally into the brain. And there are estrogen receptors expressed throughout the hippocampus. So you can see this this old but still very useful illustration of where the two canonical intracellular estrogen receptors are located in the brain. This is ER alpha and ER beta, and they're located throughout the dorsal to ventral extent of the hippocampus, as well as in the medial temporal lobe brain regions that are also affected by Alzheimer's and that feed a lot of associative information into the hippocampus. There's a third estrogen receptor that I'm not going to have time to talk about today called G protein coupled estrogen receptor, which is, its name implies, is a G protein and sits on the membrane, and that is also expressed throughout the hippocampus as well. Now, the hippocampus has been drawn the lion's share of attention in estrogens and in the field of estrogens in memory because it's really sensitive to circulating levels of estrogens. And that can be illustrated or can be shown in this illustration here, depicting the density of dendritic spines on CA1 pyramidal neurons. So you can see here, so this graphic illustrates the fluctuation of dendritic spines throughout the estrous cycle, which is the rodent version of the menstrual cycle. And during this proestrous phase, which you see here in the middle, that's when estrogen levels are at their highest. And you can see that there's substantially more of these dendritic spines on the dendrites than during the neighboring phases, estrus and diestrus, where estrogen levels are much lower. And down here in B, you can see that quantified, so that there's about a 30% reduction in dendritic spines in estrus when estrogen levels are low compared to proestrus, where the levels are high. And if we take a female rat or mouse and we remove her ovaries, we see a reduction in the density of dendritic spines and axis spinous synapses, and those can be restored with estrogen replacement. Over the years, so we've known about this effect of estrogens on the on hippocampal spine since the early 90s, and in the ensuing years, we've found that a lot of other aspects of hippocampal function are influenced by estradiol, which you'll see is abbreviated here as E2 throughout the talk. And we're specifically talking about 17-beta estradiol. So we'll talk a little bit about estradiol's effects on cell signaling, but estradiol also influences epigenetic processes and gene transcription in the hippocampus, as well as neurogenesis, various forms of synaptic plasticity, including LTP, as well as numerous types of hippocampal memory. And again, the idea here is that the age-related loss of estrogens may exacerbate the effects of aging on memory. And so for the majority of the talk, the subjects are just normal wild type C57 black 6 mice until we get to the Alzheimer's model that we're working with. And in these mice, we have found that the onset of memory decline in middle age coincides with the loss of hormone cycling. So very similar to what we see in human women. During middle age, when the cycle starts falling apart, we see the onset of memory decline. And in middle-aged and aged rats and mice, many of those memory impairments can be reversed with estradiol treatment. And I put this image here to remind me to tell you that with the exception of some data at the very end of the talk, all of the estradiol treatments you're going to see throughout the talk were delivered intracranially, directly into the brain via cannula that we chronically implant on the surface of the skull. Okay, now there are a lot of different ways in which we could measure memory in our mice. For the last, I'd say, better part of 15 years or so, we've been concentrating largely on these two object-based tasks that allow us to look at one trial, object recognition and spatial memory consolidation. So this is an example of some of the objects that we use, and we put them into a big white box. The animals are habituated to the box for 5 minutes a day for several days before training, just so they get used to the environment. And then 24 hours after the last habituation trial, the animals are trained. And by training, I mean, that we place the animals into the box with two identical objects placed near the corners. And we make the animals stay in the box until they've accumulated 30 seconds exploring the objects. And that allows all the animals to have the same amount of exposure to the objects before we do our drug treatments. Now, again, with the exception of some of the studies I'm going to talk about later in the talk, all of the drug treatments that we do are done immediately post-training. And we do that because we're particularly interested in estradiol's effects on memory consolidation. So what happens, the animals explore these objects in the absence of estrogen treatment, then they're given estrogen treatment. We know that consolidation happens in these tasks within about an hour after training. And so that allows us to look at rapid, to be able to tie these rapid molecular changes in the brain with rapid learning. And then for object recognition, 48 hours later, we test the animals. And so here, the animals are allowed to explore an object that's identical to one from training, that's a familiar object, and then a novel object. The theory here being that because mice are drawn to novelty, if they remember the familiar training objects, they should spend more time than chance, which in our case here is 15 seconds, with the novel object. The object placement task is very much the same in terms of habituation and training, we just use different objects. Post-training administration of drugs, the delay is a little bit shorter, but the real difference is during testing. So here, instead of introducing a new object, we simply take one of the training objects and move it to a new location in the box. So the key difference is what happens during testing, whereas the object recognition task allows us to assess the animal's knowledge of what the objects are. The object placement task allows us to assess the animal's knowledge of where the objects are in space. And so this is an example of the kinds of data that we see with estrogen treatment in these animals. So 30 seconds here is the maximum amount of time the animals can spend with either object, and 15 seconds is chance. That means the animals are spending the same amounts of time with the two objects, and so they're showing no preference for the novel object. That means that they don't remember the familiar training objects. And that's what we see with these delays with vehicle-treated animals. They are showing no preference for one object over the other. On the other hand, animals that receive a dorsal hippocampal infusion of 5 micrograms of estradiol do show us a significant preference for the novel object relative to chance and relative to the vehicle-treated animals. And we see the same thing in the object placement task. Again, vehicle-treated animals showing no preference, and animals infused with estradiol, in this case, spending more time with the moved object than chance or than the vehicle animals. And now we've seen this pattern of data over and over again over the last 15 years or so. It's so reliable in so many different people's hands in the lab that we use this memory enhancement basically as a behavioral probe to try to understand the molecular mechanisms that drive this enhancement. So we can ask, how does estradiol regulate memory? And there are a lot of ways in which it could do that. One way is via what are called classical mechanisms of estrogens, classical cellular mechanisms, whereby, so ER alpha and ER beta are intracellular receptors, and estradiol, being a lipid, very easily moves through these lipid bilayer membranes to get into cells where it can bind to ER alpha and ER beta. And then those hormones, that hormone receptor complex can bind to an estrogen response element on the DNA, leading to gene transcription. And while we can't rule these mechanisms out entirely as playing a role in estrogen's effects on memory, we don't think that these classical mechanisms are really what's driving them. We think that these more rapid non-classical mechanisms are what's driving estradiol's effects on memory consolidation in particular. And these would be actions at the cell membrane where ER alpha and ER beta interact with glutamate receptors or trophic factor receptors, or act just at the membrane at GPER to rapidly trigger cell signaling, epigenetic changes, gene transcription, and protein translation. And this here is a kind of a simplified model of much of what we've done so far. So everything that you see here in this model, from the receptors up here in sort of the purplish, bluish colors, the kinases in green, and the genetic and epigenetic factors in orange, these are all things that we have so far determined are necessary for estradiol to enhance memory. And I'm not going to go through, I unfortunately don't have time to go through all of these today. I'm just going to focus here on the center, and start with a cell signaling kinase called ERK, or extracellular signal regulating kinase. So ERK must be phosphorylated in order for animals to form long-term memories. That's irrespective of estrogens. This happens in both sexes, and it's true in multiple brain regions. And ERK has two isoforms, a P42 and a P44 isoform. And it's the P42 isoform that's really critical for memory formation. And around the time that the importance of ERK was being demonstrated in the literature, this is in the late 90s, there was another literature, an endocrinology literature, showing that in various cell types, including hippocampal neurons, that estradiol in vitro could rapidly increase ERK phosphorylation, P42 ERK phosphorylation specifically. So, at the time in the early 2000s, we thought, well, if estradiol is increasing ERK phosphorylation, and ERK phosphorylation is necessary for memory formation, then perhaps in vivo ERK phosphorylation is necessary for estradiol to enhance memory. And so we did a series of studies, first in young and then in middle-aged animals, and these are actually data from middle-aged animals, showing that in fact ERK phosphorylation in the dorsal hippocampus was necessary for estradiol to enhance memory. So what you see in this figure is we're looking at ERK phosphorylation measured 5 minutes after an infusion of estradiol into the dorsal hippocampus, that's IH, or the dorsal third ventricle, that's ICV. And you can see that for both of those infusion methods, we see a rapid increase in P42 ERK phosphorylation that's specific to that isoform. We never see any effects of estradiol in the P44 isoform. But what's interesting here is that when we combine estradiol with an intrahippocampal infusion of UO126, which is an ERK phosphorylation inhibitor, that inhibitor prevents estradiol not only from increasing ERK phosphorylation, but also from enhancing memory. And you can see that here. And we've gone on to show this in several different studies. And so we then became interested to know which estrogen receptor might be mediating that effect. And we thought that we decided to look at ER alpha and ER beta. And the reason we became interested in these non-genomic effects of ER alpha and ER beta were in part because a postdoc joined my lab at the time when I had moved here to UW-Milwaukee. And she had done in her graduate work, she had done some in vitro work in hippocampal slices showing that ER alpha and ER beta interacted at the membrane with metabotropic glutamate receptor 1A to increase ERK phosphorylation as well as Krebs phosphorylation. And so when she came to my lab, she wanted to see if these same interactions were happening in vivo. And so she conducted a series of studies where she infused mice into the hippocampus with the ER alpha agonist PBT, the ER beta agonist DPN, either with vehicle or with the ERK inhibitor UO126. And what she found was that, so we have object recognition on the left, object placement on the right. And what you see is that when she infused PPT and DPN with vehicle, for both tasks, we saw that both agonists enhanced memory. However, when we infused PPT and DPN in together with UO126, UO126 blocked their effects on memory and prevented both drugs from increasing ERK phosphorylation. So these data told us that activation of ER alpha or ER beta could enhance memory and that the effects of both of those receptors depended on ERK phosphorylation. Now, we also wanted to see whether ER alpha and ER beta were interacting with mGluR1A at the membrane like they did in vitro. And so we infused PPT and DPN again with vehicle or with the mGluR1A antagonist LY367385, and found, again, PPT and DPN on their own enhanced object recognition, and then in combination, the antagonist blocked their effects on memory. The antagonist also prevented both of these compounds from increasing ERK phosphorylation as well. And in some other studies that are in this paper, we did do some co-immunoprecipitations and showed that, in fact, ER alpha and ER beta are physically interacting with mGluR1A at the membrane, so that this interaction, as we hypothesized, seems to be happening in vivo. So let's see. Okay. I've gotten ahead of myself. So we then became interested in trying to figure out, well, what happens between ERK and memory? Obviously, many other steps have to transpire, and we became interested in protein synthesis because of the findings that estradiol increases dendritic spines. So I showed you that several slides ago. And if you're going to make more spines, you need more proteins to do that. And so we became interested in this other signaling pathway, mTOR, which is used to be mammalian target of rapamycin, but is now mechanistic target of rapamycin. mTOR is important in driving local protein synthesis within dendrites, and is also important for memory consolidation. Again, in theory, independent of estradiol. You see this in both males and females. And so we wondered whether, mTOR is activated by both ERK and PI3 kinase. So, mTOR seems to lie downstream from ERK, and so we thought that perhaps it might be involved in estradiol's effects on memory. And so we did, another former postdoc, Ashley Fortress, did a study where she infused mice with estradiol plus vehicle or UO126, the ERK inhibitor, the PI3 kinase inhibitor, LY294002, or the mTOR inhibitor, rapamycin, and found that all three inhibitors prevented estradiol from increasing ERK phosphorylation and enhancing memory, which told us that local protein synthesis was involved. And then we were able to, a few years later, start looking at dendritic spine density itself. And so these are just, example of our spines from vehicle-treated animals and estradiol-treated mice. So we did a study done by this was done by a former grad student in the lab, Jen Tuscher, in collaboration with Vicki Lewin at Hunter College and Maya Frankfurt at Hofstra LIJ School of Medicine in New York. And Jen infused mice into the dorsal hippocampus with estradiol, and then we looked at apical and basal dendrites in CA1, and found that we saw an increase in both types of spines within 30 minutes, and that effect lasted for 2 hours. And then what we were really interested in was whether rapid signaling for ERK and mTOR were necessary for this spinogenesis. And in fact, we found that it was. So when we infused animals with vehicle, we saw, the increase in spines. But when we co-infused animals with estradiol plus the ERK phosphorylation inhibitor or the mTOR inhibitor, both inhibitors prevented estradiol from increasing spines, telling us that, this was really exciting for us at the time because people had known, that estradiol increases spines since the early 90s, but we didn't know how that happened. And so this was the first study to show that rapid activation of cell signaling kinases, in this case, ERK and mTOR, was necessary for estradiol to increase dendritic spine density. So that was really exciting. There are some other really cool findings in this paper that I'm not going to have time to go into today, where we showed that estradiol infusion into the dorsal hippocampus also increases dendritic spine density in the prefrontal cortex. And we subsequently done studies using some chemogenetic manipulations to show that, in fact, the dorsal hippocampus and the medial prefrontal cortex need to be active simultaneously in order for estradiol to enhance memory. But that's kind of a story for another time. And At this point, I could go on about some of our additional estrogen studies. We've done some recent work that's in its sort of early stages, showing that not only is protein synthesis important for estradiol to enhance memory, but protein degradation is important. And this is particularly degradation via the ubiquitin proteasome system. And then we have started to look outside of the cell, outside of neurons, at what's going on in the extracellular space. So we have some new data that's showing that matrix metalloprotease 9 out in the extracellular space is critically important for estradiol to enhance memory, as is BDNF binding to TrkB receptors. And interestingly, there's MMP9 can cleave pro-BDNF into BDNF. So there's an interesting link between those two studies. And then we're just starting to look at astrocytes as well. And we have some new data showing that glutamate transporters on astrocytes are important for estradiol's effects on memory. And we've got some work going on looking at lactate usage in neurons as well. So, but instead of going into more basic research, I thought I would transition to some of our Alzheimer's work, since this is an Alzheimer's initiative. And so moving on to kind of the second bubble in our model. So I'll take us back to the initial sex differences that drive the research, and just remind you that for late onset Alzheimer's disease, there are several risk factors, of course, chief among them being age, another being female sex, as I've already mentioned, and then, the other is ApoE4 status. And so we've been interested in apolipoprotein E, which is a lipid-carrying protein involved in cholesterol metabolism and transport. And it transports, a lot of things into neurons. It transports A beta into the cells for disposal, as well as cholesterol into cells for conversion to estradiol. And the brain does make its own estradiol. And we and others have shown that this brain-derived estradiol is actually critically important for memory consolidation in both males and females. As you, I'm sure, are well aware, there are three alleles of ApoE, E2, E3, and E4, where E2 tends to be protective or lowers one's risk of Alzheimer's. 3 is neutral in terms of risk, and 4 increases one's risk. And 4 is the least effective at transporting A beta into cells for disposal, so is associated with inefficient, or it's associated with neurodegeneration. And ApoE4 carriers have more and earlier appearance of plaques, of tangles, phospho-tau accumulation, brain atrophy, and cognitive decline. And again, as I'm sure you're aware, women who have 1 or 2 copies of ApoE4 are at greatest risk of developing the disease, particularly women who are homozygotes for carrying 2 copies of E4. So women who are E4 carriers are at greater risk of developing the disease than men who are E4 carriers, or men or women of any other ApoE combination. And some clinical data suggests that estrogen therapy is actually less beneficial cognitively for women who are E4 carriers. And we think this could be because of the reduced ability of ApoE4 to transport cholesterol and dispose of A beta could contribute to increased risk of Alzheimer's disease in women in a number of different ways. Number one, it could reduce estradiol synthesis in the brain. It could reduce estradiol-induced neuroprotection, increase A beta and phospho-tau accumulation, and promote synaptic and neuronal degeneration. So how do we mitigate this increased risk in women? One way is to work with model systems. I'm fully aware that there is no good, model for Alzheimer's disease, but we're working with one that I think is pretty good. Well, number one, with these mouse models, we can ask, why do sex and ApoE genotype effects exist, and why do double E4 females fail to respond to estrogen therapy. Because if we can address those in our mouse models, then perhaps we can develop treatments to mitigate the effects of sex and ApoE4 on risk and perhaps increase estrogen responsivity in women. So we've been working with this 5X FAD ApoE transgenic model, we call it the EFAD model, that was developed by my colleague Mary Jo Ledoux at the University of Illinois at Chicago. And MJ's mice, so there's 3 APP mutations, these listed here, 2 presenilin mutations, and then they also express human ApoE2, 3, or 4. And the E4 FAD mice, so these animals have 2 copies of E4, they're homozygotes, have impaired spatial memory as well as, you'll see, object memory. They have accelerated A-beta accumulation. And as you can see in this figure, they have more A-beta 42 as well as more oligomeric A-beta in the cortex and the hippocampus. And we can see these effects by 4 months, but they're really particularly striking at 6. So that's the age at which we've been working with them. And what's nice about this model is that the pathological changes are greater in the E4 females than they are in E4 males, or than E3 mice of either sex. So it recapitulates some of the sex differences that we see in humans. So we've conducted a couple of studies with, well, we've conducted now several studies with these mice. We've just published the results of 2 of them. I'm going to show you, these data, we've published, I should have put this in there for Christina, but I didn't. I'm sorry. We just published some data on anxiety in these mice, but I'm going to show you the memory and spine data. So these data show you the effects of sex and ApoE genotype on object recognition and object placement. So a couple things to point out here. Again, these are mice that have 2 copies of E3 and 2 copies of E4, males and females. These animals, we tested the animals in this study at shorter time points than I've shown you before, only 24 hours for object recognition and 4 hours for object placement. We did that for 2 reasons. Number one, we didn't think these animals would remember as long because of the Alzheimer's pathology, but we also wanted to give them a chance to show intact memory. We use the longer delays for the estrogen treatment experiments because those are delays at which we know that our vehicle-treated animals will not show memory. And so we can see an enhancement of memory by estradiol. In this case, we strongly suspected that animals will be impaired. And so we use these shorter time points. And you can see that the male E3 animals do learn at these shorter time points. So they're showing memory relative to chance for both the object recognition and the object placement tasks. But none of the other groups showed intact memory. So both male and female E4 animals, as well as female E3s, were impaired in both of these tasks. And you can see for the object recognition task, not only are they impaired relative to chance in that they're not showing any improvements relative to chance, but the E4 animals are impaired relative to the male E3s. So this is kind of interesting. We expected, or we thought that we might see a sex difference in the E4 animals, but we think that perhaps we had waited to test them too long. We might have seen a difference between males and females if we tested earlier, but we think, at 6 months, the pathology was so extreme that both males and females were impaired. Now, in terms of spine morphology, we looked in both the CA1 region and the hippocampus, as well as the prefrontal cortex. And I'm just going to show you CA1 because the prefrontal data are exactly the same. We looked not only at total spines in this case, but at spine type. We looked at thin, stubby, and mushroom spines, with mushrooms being considered the most mature spine type. Stubby is intermediate and thin is immature. We did see a reduction. So here we see just genotype effects, that the E4 animals of both sexes have lower spine density in terms of total spine density. They also have fewer mushroom spines and fewer stubby spines. There were no genotype effects on thin spines. So here, these E4 animals have a reduced number of these more mature spines, as well as the intermediate spines. We also use Western blotting to look at some proteins. We looked at the presynaptic protein synaptophysin and the postsynaptic protein PSD95. And found, again, genotype differences here, no sex differences, but that in the E4 fat animals, we see lower levels of PSD95 and synaptophysin, which is consistent with the spine density data. And then we did see a little bit in terms of sex differences in some other proteins. So glial fibrillary acidic protein, or GFAP, was an astrocytic protein. We do see that female E4 fat animals have the highest levels of GFAP, suggesting perhaps more astrogliosis in female E4 animals. The female E4s also have the lowest levels of Krebs phosphorylation, suggesting perhaps decreased transcription. And interestingly, we saw a genotype effect, no sex difference here, but both male and female E4 fats have more ER alpha. They have no differences in ER beta, but they do have more ER alpha, which we think of potentially as maybe a compensatory mechanism. They're trying to ramp up ER alpha to try to be able to capture whatever estradiol is available to stimulate cell signaling. Now, in a subsequent study, we took just females and treated them with estradiol. So here we're looking at not only the E3 and the E3-4 animals, but in the E4 animals, but we've added a heterozygote group and 3-4 animals. So again, these are females, these are overectomized. The previous study, all those animals were gonadally intact. But here we wanted to look to see whether we could replicate the clinical finding that women who have 2 copies of E4 respond more poorly to estrogens. And we did do that. So here we can, this is again, an acute treatment with estradiol immediately post-training in our object recognition, object placement tasks, and find that the E3 animals and the E3-4 animals are responsive to estradiol, but the animals with 2 copies of E4 are not. And we see that same thing mirrored in terms of apical spine density, total spine density. So estradiol increases spines in the E3 animals and the E3-4 animals, but not in the E4 animals. So the next question for us to try to answer is, why do those E4 fat animals become unresponsive to estradiol? So that's an open question that, but we're hoping that the answer will provide us some useful avenues for potential drug development. So let me then touch in the last few minutes of my talk. I think I'll be able to make up for a little bit of lost time to tell you about some of our work on drug development. So I've been working for several years now with a couple of colleagues here in Milwaukee, a synthetic chemist, Bill Donaldson, who's at Marquette University, and a pharmacologist, Dan Sem, who's at Concordia University of Wisconsin, to develop selective ER beta agonists. So as you recall from the beginning of the talk, if we stimulated ER alpha or ER beta, we could enhance memory in young animals. And we've been focusing on ER beta because many of the detrimental side effects of estrogen therapy in women are due to estrogen's effects on ER alpha. So stimulating ER alpha increases cell proliferation in breast cancer cells, but not ER beta. ER beta actually seems to be anti-proliferative. And ER beta has beneficial effects on memory, as well as on depressive and anxiety-like behaviors. And so we think that perhaps targeting ER beta for menopausal women could provide benefits for memory without some of those side effects that are associated with stimulation of ER alpha. So Bill has synthesized a library of probably well over 100 compounds now, but we've been focusing on this particular compound we call EGX358, which you can see here, its structure in red overlaid on the structure of 17-beta estradiol. It binds ER beta with very high potency and extremely high selectivity. It's 750-fold more selective for ER beta over ER alpha. And as comparison, DPN, the compound that I showed you we used earlier to stimulate ER beta, is only 70-fold more selective for beta over alpha. In our assays, we show that it doesn't bind to any other nuclear hormone receptors. It doesn't have any effects on metabolism or breast cancer cell proliferation or some gross tissue pathology. And so several years ago now, a former grad student, Jae-Kyun Kim, did a series of studies where he treated animals with EGX-358 in multiple doses in a variety of different routes of administration. So we started with a dorsal hippocampal infusion. We treated animals with vehicle, DPN as our sort of gold standard ER beta agonist, and then multiple doses of our experimental compound, EGX. We tested these animals in both object recognition and object placement, and I'm just showing you data for object recognition because the object placement data are identical. But you can see here that multiple doses enhanced memory in this task. We also treated these animals with an intraperitoneal injection and found a similar memory enhancement. So then we moved to oral treatment and moved to oral gavage and found that multiple doses enhanced memory via oral gavage. So then we wanted to see, now these were all acute treatments, again, immediately post-training, but that's not the way that women take estrogen therapy. They take it chronically, they take it every day. And so we wanted to see what the effects of long-term treatment might be on memory and a variety of other things. So in 2018, my colleagues and I formed a company that's called Estrogens Therapeutics, for which I'm the chief scientific officer. And we went through a National Science Foundation entrepreneurship training program that has fledgling companies talk to potential customers and potential users of their product. So we went out and we talked to physicians and menopausal women to see if, our initial hypothesis was that women would pay for a drug that would decrease their risk of developing Alzheimer's disease in the future. Well, it turns out when we talked to people, many women and even many physicians weren't necessarily all that aware of the fact that they were at greater risk of developing Alzheimer's disease. Really, for many women, memory loss wasn't, or at least risk of Alzheimer's disease wasn't on their radar at all. They were more concerned with immediate symptoms like hot flashes and mood swings, anxiety and depression. Although some women definitely do suffer with memory loss in the early stages of menopause, and many of them have reached out to us for help. So, but this caused us to retool a bit in my lab to start measuring hot flashes and anxiety and depression. And this work is done by a current grad student in my lab, Aaron Fleischer, who did this heroic study where he did long-term EGX treatment given by oral gavage every day, and then he tested them 1 hour later through a series of tasks, as you see here. First, he looked at hot flashes by measuring tail skin temperature, and I'll mention more about that in a minute. Then he ran them through a couple of anxiety-like behavior tests, open field and elevated plus maze, a couple of depression-like tests for tail suspension and forced swim, of course, our memory paradigms, and we measured, we had body weight measurements that we analyzed because, of course, we were weighing the animals every day for drug treatment or drug dosing. So this is the experimental design. The animals were all overrectomized, and then we started treating them with our compounds. They got treated with vehicle, estradiol, DPN again as a positive control, and then the lowest effective dose of EGX-358. And they got treatment for about 2 weeks before we started doing anything with them. Then we looked at, I'm going to show you data from tail skin temperature, memory. I'm going to skip over the anxiety and depression task because we didn't see anything there for any of the treatments, actually. Well, we saw a little bit with estradiol, but not with our experimental compounds. So I'll talk about memory and then body weight at the end. But the animals were treated in total for 64 days. So we had to figure out how to induce and measure a hot flash in mice. Hot flashes are sudden onset of it's a rapid increase in temperature that is very transient. And while overectomy can increase baseline body temperature in mice, it doesn't cause these transient episodes that a hot flash is. So we started inducing hot flashes with a drug called Sanctide, which is a neurokinin 3 receptor agonist that causes a rapid increase in tail skin temperature. So we do infrared imaging of our mouse's tails, and this is on top, a mouse injected with vehicle, and you can see the tail is nice and cool. You can kind of barely see it. And then in the bottom, this is an animal injected with Sanctide, and you can see a red hot tail, because the animals don't they don't pant and they can't sweat. So the tail, they release heat out their tails. It's like their radiator. And these are data. So on the top, you see we have these 4 groups of animals that are injected, that were gavaged every day with vehicle, estradiol, DPN, or EGX. And then we challenge them with Sanctide or vehicle. And so on the top, you see our vehicle challenge, and you can see that the animal's body temperature increases a little bit when we pick them up and inject them. That's the y-axis. But then body temperature rapidly goes back down to baseline. In the bottom, you see animals treated with Sanctide, and you can see here a very rapid increase in tail skin temperature and a very rapid decrease. And in black are the vehicle treated animals, and you can see that the tail skin temperature is higher. It's significantly higher in those animals than in any of the estrogen treated animals. So for our interests, EGX is doing just as well as estradiol or DPN at reducing tail skin temperature. In terms of memory, we see the same thing we did with our acute treatments, that again, in terms of both object placement and object recognition, all 3 estrogen treatments are enhancing memory. We also saw, like I said, no effect on anxiety-like behaviors or depressive-like behaviors, no effect on body weight, which you see here. I mean, everybody increases body weight after an overectomy. That's just what happens. But there were no significant differences here. So, in a glass half full way, we find, maybe it's unfortunate that there's no beneficial effects, but there are no detrimental effects of this compound on mood or body weight, which we think is a win. So, and then the final piece of data I'll show you is our most recent data. We got funding from the University of Wisconsin system to actually look at the effects of EGX358 in our EFAD Alzheimer's mouse model. And so we did a very similar study. Aaron collaborated with another grad student in the lab, Miranda Schwab, to run them through basically the same experimental design I just showed you. Here the results, we're still analyzing the results, we're still analyzing the brains from those animals, but so far we see promising effects in the object recognition task, where our novel ER beta agonist is increasing or is enhancing memory in the object recognition task. In this case, we just have E3 animals and E3-4 animals. We could only afford to do 2 genotypes because these animals are really expensive to produce. And we didn't think we'd see anything in the E4 animals based on our estradiol experiment. So, but in these 2 genotypes here, we are seeing beneficial effects of animals. And these are animals that already have pathology. So maybe that's promising in terms of patients who already have Alzheimer's disease, but maybe that's just wishful thinking. So anyway, I've shown you some data that address each of these 3 strategies for trying to reduce memory dysfunction in older women, and hopefully, EGX or similar compounds. We have a couple of other potential leads that are actually even more selective than EGX358 might find their way into clinical trials for menopausal women that may have benefits for Alzheimer's risk at some point in the future. So I'll end by just acknowledging the folks who've done the work. So everybody whose name here is in bold and italicized, or somebody whose data I showed today. I want to thank our funding sources. And if you want to, reach out to me, feel free to send me an email or reach out via social media. So thank you very much. I'll stop sharing and I'm happy to take questions.
Thank you, Karyn. Thank you. It was really interesting and exciting, of course. And I wish we had more time to hear about the rest of the data, because I know there are so many. And I have a few questions, but maybe first we should ask the audience.
Sure.
Do we have any question from the audience?
Maybe I can start, Christina.
Yes,
Thank
please.
you very much for this excellent talk and extremely interesting. I was wondering about if there are any estrogen receptors in microglia and what's going on with microglia activation, what's the role of estradiol in microglia activation and neuroinflammation, and if there are any differences between in terms of sex, you know.
That's a really good question that I don't know the answer to. We haven't looked at microglia. In terms of non-neuronal cells, we've been primarily focusing on astrocytes, which do express all three estrogen receptors. I don't know, Christina may know more than I do. I certainly suspect that microglia do have estrogen receptors, and there are, our colleagues, Peg McCarthy and many others have been studying microglia, but I just don't feel like I know enough about them to say anything particularly credible.
It's okay. Thank you.
Sure.
Yes, that's also a very interesting line of research.
Yeah.
They're very important during development, but also it seems in the adult life as well.
Yeah, since neuroinflammation is a big thing, especially, and we are start exploring this in Alzheimer's disease, I was wondering.
Absolutely.
Yes.
Yes, I mean, neuroinflammation, microglia, complement signaling, obviously have been increasing in has gotten a lot of attention recently. So I think there's a lot to be learned about neuroinflammation and its role in Alzheimer's. So.
Thank you.
Yes, actually, my question is about the GPR1 receptor, the brain receptor, if you have any data on that, and how it mediates these effects in memory.
Oh, for sure. Yeah. So I know, I could have gone on about that too. So we have, so GPER is a really interesting receptor because it was an orphan G protein coupled receptor until fairly recently. It does bind to estradiol in certain cell types, and so its name was changed several years ago. And we have been using a pharmacological strategy to look at GPER function. So if we treat animals with a GPER agonist, it enhances memory just like estradiol and just like PPT and DPN. And initially, we figured that it probably worked by activating ERK, just like ER alpha and ER beta, but it turned out that it didn't. So when we treated animals with UO126, the ERK phosphorylation inhibitor, it did not block G1's ability to enhance memory. And through a series of studies, we just found that estradiol is going through ERK and influencing memory, but GPER influences memory through a different MAP kinase called JUNK, C-Jun N-terminal kinase, and that there doesn't seem to be any communication between estradiol and G1 signaling in terms of enhancing memory. I don't know why that is, that in our hands, and we've seen this now in several studies, so I believe it, and I don't know why GPER doesn't seem to be acting like an estrogen receptor, at least in the hippocampus. And again, with our parameters, other colleagues have seen, that GPER can influence ERK phosphorylation in other ways, but GPER also increases dendritic spine density, and it seems to do so through actin signaling. Not actin signaling, cofilin signaling, which leads to actin polymerization, or cofilin phosphorylation leads to actin polymerization. So that's a really interesting story, but it seems to be a very different signaling pathway that regulates the effects of GPER than ER alpha or ER beta.
Yes, that's very interesting, because we are also running some studies with GPER1, but for depression and anxiety. So we'll see if it's the same. Hopefully, so. Yes, okay. Thank you very much. I think if we don't have other questions, maybe we should finish. Do we have any questions on YouTube?
Right. I certainly appreciate, we've gone over time. So
Yes, but thank you very much again for being with us.
Yeah, you're welcome.
We hope we'll be able to do it again, hopefully not virtually. Maybe
Yes.
we'll invite you in Greece at some point.
That would be
We
wonderful.
can all visit.
And
Yeah.
also in Baltimore, I also want to thank you on behalf of HIAAD for your accepting the invitation and for giving this excellent talk today. Thank you very much.
Thank you all. Great to
Thank
see you.
you. Have
Great
a
to
good
see you.
day. Bye.
Bye-bye.
Bye.